Hello, welcome to a short series of videos on how to use PyMOL. PyMOL is a structural visualization tool which is used to look at the structure of various protein complexes, DNA or RNA. So first we're going to start off with the structural techniques used to generate these files. So the first is X-ray crystallography. This represents almost 90% of the PDB, but the problem is it requires a protein crystal to grow. Proteins have to be structured. Any dynamic regions will not be crystallized. But the good thing is with X-ray crystallography, there's no protein size limit. So what you do is you start off with a crystal, you hit it with a load of X-rays, which then generates a diffraction pattern, and from there you can calculate the electron density, which then you use to fit your model. Second method is NMR, represents about 8% of the PDB. The great thing about NMR is your protein's actually in solution, so no crystal required, and it allows you to do protein dynamics. The principle of NMR uses the fundamental property of the atom, which is its spin. However, it does require labeling the protein, which can be costly, and you're only limited to a relatively small protein size. The last method is electron microscopy. This represents 3% of the PDB. However, this is growing year on year as technology in electron microscopy has improved so much that we can now capture atomic resolution structures. There is a size limitation. The bigger the sample, the better. However, this limitation is getting smaller and smaller every year as technology improves. The principle of electron microscopy is like a light microscope but using electrons instead. You take a picture of your sample, which is frozen. You then cut out each individual one and then align them to generate 2D classes, and from there the software will orientate these 2D classes to form 3D classes, which then you can then generate an electron map. The PDB. The PDB is the database where all structures are saved. So if someone solved a protein structure, it will be uploaded to the PDB. And from here, anyone can download files from the PDB, and it's all free. The PDB looks like this, where you have this 4-letter/number code, which is unique to every structure in the PDB. And it has information about the protein is in there, the organism it's from, when it was deposited, the paper it's associated with, if there's any macromolecules, so these are the actual proteins, and any small molecules such as ligands. But you can't always trust the PDB. The PDB will always provide a validation tool which will give you an indication of how good the structure is. The further these are to the red, the worse they are. So the clash score is when two atoms are too close to each other. Ramachandran outliers are when the angles of the backbone of the psi and phi angles do not fall into the correct angles, and then side chain outliers as well. The further to the blue it is, the better it is. This sort of represents the average. The average at the same resolution that this structure is at. So now on to PyMOL. The reason we use PyMOL, because if you were to actually download a file from the PDB and open it, all you'd see is this. This is basically a text file which contains all the atoms and their coordinates, which is not very useful. PyMOL allows you to visualize that in a really easy and simple way. It's free to all students. You can download it from the PyMOL website. If you go to Google and type in PyMOL Educational, there's a unique web page which you put in your email address and which university you're at, and it allows you to have an educational license which lets you use this for free. It's open source. It's constantly being developed. You can use this on either Mac or Windows, and the current versions of Mac and Windows. You can view any sort of protein, DNA or RNA. It's easy to use. It has quite a lot of tools, so it's more difficult to use than something like RasMol, but RasMol has not been updated for a very long time. So PyMOL is up to date, which means they've made it simpler. In the next video, we'll go to the actual PDB website and we'll download a structure. The third and fourth videos will be actually using PyMOL. The third video will be just what all the buttons do. A fourth video will be using some of the more complicated tools.